Okay. Yeah, thank you. In Rosalind Allen's lab from Jena University, I'm also part of this cluster of excellence balance of the microverse. So I'm not asking you to do any morning fitness this time anyway, but we are gonna stay anyway in the seawater because I'm gonna present you something regarding the dynamics of algae-bacteria interactions in artificial plankton communities. So a work done in my lab in collaboration with the lab of Georg Ponert, that is still another lab in Jena. So first of all, something regarding the players, phytoplankton. Phytoplankton is, as you see here, this is a sea drop under the microscope, so it's quite densely populated. You see a lot of nice colorful guys, and phytoplankton is composed by unicellular algae and the bacteria that are living with these algae. Why is important? Because it fixes up to 40% of the global CO2 into nutrients, and this is also the fuel for the marine food web. And the phytoplankton has a nice structure over time, so tends to bloom during springs, and after goes on by dying a little bit and regrowing, and starts again in springs. So it has a cycle, and this cycle is also possible to be monitored in the lab, because, for example, here you see a specific alga that is called diatom, and measuring the chlorophyll in the system, you can actually see whether the system is in an early growth stage or in a late phase. And so you can actually, by monitoring the chlorophyll, plot in the amount of algae that is proportional to this chlorophyll, and knowing over the days how they changes. So since it's so important for the fixation of CO2 and the in general the geochemical ecology, it's also important to understand the diversity of this phytoplankton, the dynamics, and the stability. So we are going to focus on actually only one of these topics, but also the other one are obviously important for understood the system. As I told you, it's a complex system. It's made up by algae that interact with some bacteria, and they can actually interact in a positive way by exchanging, for example, carbon sources or amino acids or other nutrients. But they can also have some negative interaction. For example, there are some bacteria that can release some toxic compounds that decrease the growth of the algae. Obviously, the growth is determined by the community composition and also the interaction among these players. If you are in the lab, you can actually simplify the system by thinking that you have a bunch of algae here and some bacteria that are interacting with them, and we have to understand how these interactions are specified over time, so given the dynamics of the interactions. And there are too many questions that I would like to answer during this talk, and they are how bacteria interact with algae and affect the dynamics of a community. So what is the interaction between specific groups of bacteria and the specific algae, whether they are positive, negative, or maybe neutral, and whether specific bacteria are going to play a universal role in the interaction. So what does it mean universal? Means that maybe the same species of bacteria are always behaving equally over the whole algae growth phase and late phase, or maybe some bacteria are behaving differently in different phases, or even if some bacteria are behaving specifically in some behavior with a specific algae. So how we do that? Actually, that's our research methods. And first of all, we screen for relevant bacteria-algae interactions. So we chose several algae and several bacteria that are living with them in the natural environment, and we first of all understood how they behave and chose some of them. So once you have done this screening, we first of all understood how the growth curves are done in isolated culture. So you grow alone diatoms and grow alone several species of bacteria and check the growth culture. So pretty easy. And after you do pairwise interaction, I mean, that's the standard way of understanding the initially the interaction between species. So you group the algae with a specific species of bacteria, and you check how they grow together. And from that, we try to develop a mathematical model that was able to predict, hopefully, the community dynamics. And actually, I will show you how to do it. And finally, we created in the lab an artificial community composed of a specific algae, the diatom actually, and 4 different bacterial species that we chose. And by using the mathematical model, we try to understand how this system develop over time. And obviously, we would like to compare the experiment to a mathematical model prediction. So that one is basically the outline of the talk. So now I will guide you through this kind of procedure. The work in the lab was done mainly by this guy, Yun Deng, a postdoc in Georg Ponert lab. And what it did first for screening the bacteria and the algae was to take, obviously, a bunch of algae and a bunch of bacteria that usually live together in the natural environment, and to check, first of all, the growth of the algae alone. So you can monitor the chlorophyll, as I told you, over days. So there is an exponential phase, and usually after a late or a death stationary phase, depends on the algae actually. And you add bacteria in the same environment, and you check how this growth curve changes. And to be fast, it just took 2 points, one in the exponential phase and one in the late exponential phase. Sorry, in the late phase. And he compared the two actually. What he got is that you have a matrix of interactions during the early growth phase and the late growth phase. And if you see positive interaction, you have a yellow color, and for negative interaction, you have a blue color. So positive means that when you add the bacteria, the algae grows better. And when you add bacteria here, if you see blue, means the algae is repressed in growth. So as you see, here there are the different bacterial species that you use, and here are the different algae. The matrix is quite scattered. So that one is already an indication that there is no universality. So the effect of a given bacterium depends strictly on the algal partner. But you can also see that if you would like to zoom a little bit more in this map, there are patterns that are completely non-conserved. So for example, this one that was completely blue here gets yellow and so on, but there are actually many. I highlighted so only a few of them. And this one is an indication that there is a growth phase dependence. So bacteria have often diverse effects on the algae that are different from the early and the late growth phase. So it depends on the growth phase that you are into. So this is also an indication for the modeler that if you like to model this system, you have to divide at least in 2 pieces your structure. So in the end, Jung decided to use, by checking the screening, some bacteria that are living usually in the natural environment with this diatom that is called Cosinodiscus radiatus. And this bacteria, I will call it for short with some names because I cannot remember all of them, they're complex. CS4, Rose1, Croce1, and CS1. And actually, I mean, I'm sitting in Jena, and that's a nice place to be because Ernst Haeckel was one of the guys that gave a very nice drawing of these diatoms. Actually, here in the plot, in the picture, you cannot see the diatom that we are using, but they are very similar. And this is a more modern picture of these kind of diatoms. Actually, we have also our diatom that we are using, this Cosinodiscus radiatus. So what Jung did at the beginning was pretty simple in theory, not simple in practice, because it's a complex experiment indeed. He took some culture of the diatom, Cosinodiscus radiatus, and he checked the growth rate over many days, and counted by eye, actually by hand, in the microscope the amount of diatoms that you have in the environment. And if they are growing alone, you see this orange line that is actually the same in every plot. And after you input together the bacteria with the diatom and check again the growth of the diatom. So the colors here are the co-culture of bacteria and diatoms for the diatom counting. So here for CS4, cross A1, rows A1, and CS1. And did the same also for bacteria. So for bacteria, the experiment is basically the same. So you grow bacteria alone, and you see the growth of bacteria given by the gray lines here. So for different bacteria. And with diatoms, you counted again the amount of bacteria in the system, and you have these colored lines. Actually, so you can see that some of them are really changing. Both from the exponential growth phase and the late stationary phase. So they are not keeping fixed the behavior. And especially you can notice that, for example, this guy is the presence of CS4 bacteria is increasing the growth of the algae in the late phase. Also, this guy is doing the same. This guy is doing the opposite, so repressing the growth of diatom in actually in both phases, I would say. And this one is doing something very strange. So it's shifting the initial starting point of the growth of the diatom on a later stage. So it's inducing a lag phase in the growth. And notably, this guy, Crosey, doesn't grow without algae, but in the presence of algae, it grows very nicely. So what can we work out from these simple ideas? That each bacteria diatom pair has a different growth dynamics that depends on the algal aging. And to understand these dynamics, we created this mathematical model, very simple, Lotka-Volterra. So during these days, we heard a lot about Lotka-Volterra, pros and cons. But it's actually the first approximation to understand what's going on in a system, because it tells you how the cell density and the different subpopulations that are interacting in the system changes over time. So here we have actually the diatom and the 4 bacteria. That depends obviously on a growth rate, on a self-interaction term, and the cross terms that are giving the real interaction between the different populations in the systems. Obviously, if the cross terms is positive, we have a positive interaction, negative interaction, and for 0 means that is a neutral interaction or no interaction at all. So how we fixed all the parameters in the system, very simply using what you have seen before here. So growth curves alone in isolation, and growth curves in combination. And from the growth curve in isolation, we obtain the growth rates and the self-interaction terms that are expressed in basically the fight for nutrients. You could. What about bacteria interaction? Yeah, well, we come to that. Yeah. And after, you can use also the pairwise culture to obtain the crossing structure, so with AJ terms. So basically, you see here we have a full system that is complete. And if you would like also to understand how it's going on, we can draw the interaction we obtain from the fit in a diagram. So most of them are actually negative. There is this one that is very strongly positive. And we know it already from the plot because it was this guy here, obviously is really positive. And this guy is actually the guy that shifts the starting point of the growing culture of a diatom. So coming back to the presence of the higher interaction terms, they are not present in the model at this stage, and I will show you why we didn't put them. Obviously, as I told you, we have different stages, so different phases. And we use 2 sets of parameters depending on the algal phase, the early exponential phase, and the late stationary phase. So that one is actually the last slide. I will go in detail. Once we have all the pairwise interaction, we can put together all the system and try to reproduce what the experiment with a diatom and the 4 bacteria growing together are given us from experiments. Just to remind you, here you have the diatom cell density and here the time in days, and that's the case that we fit to obtain the parameters for the algae. So basically, it's the diatom that is growing in isolation. In dashed line and dots, you see the experimental part, and the fit in solid line. That's actually just the case that you should compare when you grow all together. And if you grow all together, you obtain this dashed line with the dots that represent the number of diatoms that you have in the system. And the model that I presented you before, so this one, gives this nicely agreement with the experiment given by this solid line. So coming back to the question regarding the higher interaction, already at this level, we have a nice agreement even without higher interactions. You can also say that error bars are quite large and we cannot actually distinguish interactions that are quite weak. So at this stage, given the error bar that we have, we don't need to put in the system higher interaction. If you would like to know whether we tested them in the lab, no, that one was not done. So we are not sure whether the errors were very weak. But at this point, actually, we cannot really see them, or at least distinguish them. So indeed, pairwise interactions are sufficient to explain the behavior at this stage. And if you would like also to compare the system with the neutral case, so basically the case in which you don't have, you sum up all the components independently, that one would be given by the green line. So means that the interactions are important. We cannot neglect them because the system would behave completely differently. And there is another case to take into account, and is this one. So if you check at the strength of the interactions, most of them are very, very weak, and actually you can neglect them. Only these two are important, the effect of the diatom on the Crocei bacteria, and the shift given by the CS1 that is actually difficult to put in the Lotka-Volterra model because it's not designed to put leg phases. You have to play a little bit. Actually, we divide the system in another sub part at the beginning that gives this leg phase, so one more phase, let's say, to fit, and that's why you see this kind of kink here. And if you just take into account these two interactions, you already be able to catch quite properly within the error bars the system that is given by this magenta line. So just keeping these interactions that are the leading interaction, the dynamics is quite nicely explained. And with that, I would like to summarize. So what we have seen here was a study of the algae-bacteria interaction in phytoplankton. Interaction depends on the bacterial and algal species, so that's important. Bacteria don't show any universal growth promoting or inhibiting effect on the diatom, so there are bacteria that are promoting growth during the first phase and repressing in the late, or vice versa, happens both. Community dynamics depends on the algal growth phase, as I already told you. And as an outlook, obviously, there is the question why we have this kind of interaction, what is positive, what is negative, and why is positive or negative. And so we would like to investigate complex communities that are more similar to the one that you can actually find in nature, although this one was already at this level, at this stage, designed in such a way that all the density of bacteria and algae were really similar to what you can find in a seed drop, in a natural one. And obviously elucidate the factors that are promoting or inhibiting this bacteria-algal interaction. So there are some nutrients, amino acids, vitamins, carbon sources, because we know that diatoms are releasing carbon sources used by bacteria, how they interact. And obviously there is this puzzle of CS1 that is shifting the initial growth. So probably there is a toxic compound released. We don't know how and what. That one would be the next. And with this one, I would like to, first of all, acknowledge the people that worked in this project. Yoon, that did a great job in the group of Georg, Rosalind, my supervisor, the people that found us. And if you're interested also in theoretical biophysics, so more on the modeling side, you can follow us in Twitter because we have a Twitter channel. And we have also a podcast. So if you'd like to be part of a podcast or listen to us, I mean, just subscribe. We have also a virtual seminar series online. You can subscribe for free. We are on Zoom every 3 weeks. And we have a good, nice set of speakers. Some of them are actually here. And finally, if you're interested in the work, you can just check the paper that is being accepted and in press. So Beton, I would like to thank you, all of you.
Thank you for this nice talk. Are there any questions? There's one there.
I just want to ask you about so you said you have two sets of parameters for the Lotka-Volterra.
Yeah.
And I mean, you showed nicely that only these two interactions matter. And I was wondering also what's the difference between using one set or two sets of parameters?
Yeah, okay, so the two sets of parameters are basically, the first set is for the exponential growth phase and the second one for the late phase. Actually, you can see them here. So these are the two sets of parameters. I mean, probably doesn't give you much information, a table with numbers. But what's happened is very simple. In the first phase, you have all the the bacteria that are actively growing and doing something with the algae. In the second stage, it's much easier. All of them are actually doing nothing, they are stationary. There is only algae that are dying, so there is a negative growth rate here. And there is one guy that is, I will show which one actually, this one that is interacting with the algae. So first set, all of them active and doing something. Second set, only the rosé one that is influences positively the algae. So the two sets are completely different. Basically, we have completely different equations.
So the second set.
Yeah, exactly. So the main effect is the death of the algae, because actually we don't know whether there is an effect of algae that are finishing the nutrients. Probably not, because we tested a little bit. But algae are actually dying because of aging. So that's everything. That's the main effect.
Other questions?
So I was wondering whether this effect you observed in the lag phase, if that could be an experimental bias for that particular population. You know, it's just that it's particularly susceptible to the changing environment from the medium where they grow before mixing with others.
No, they tested actually that one. Also giving for, I mean, longer time for adaptation. And no, apparently there is a real shift.
In the behavior when you mix with others.
Yeah, they also did a screening regarding the kind of compounds that are released and taken up by both algae and the bacteria. But at the moment, we cannot give much insight on it. We know that some bacteria are deficient of some amino acids and vitamin, especially vitamin B, and so that are taken up by the bacteria given by the algae. And we know also that bacteria need some carbon sources that are released by the algae, specific one. But we don't know what's going on there for the CS1. It's something that they think that is actually a toxic compound because they have looked at the genome and they've seen that there are some specific genes that are connected to that. So might be the case, but it's not a lag phase due to adaptation in a shift of nutrients. Actually, the nutrients are the same, so it should not be there.
Did you try to put a delay in your model in the interaction
Yeah.
term to see?
Yeah, I tried. I mean, it's not super easy, obviously. If you would like to put a delay, you have to think about how to do it also. I mean, there are several ways. You can have subpopulation that is growing and one not growing. You can put a delay like a compound that is repressing a toxic one, or maybe actually a delay like I did in dividing the model in one more piece for the CS1. Actually, the simplest, obviously, that I presented here was dividing the model in several pieces. But I also tried with this toxic compound, unknown toxic compound, and it works nicely anyway. So might be that is a compound, but I mean, it would be speculation at this point, so I cannot really say it works, but I cannot present it.
Short question, short answer.
Yeah.
Yeah, I'm wondering, the environmental conditions may really modify these interactions. Are you going to play with changing environmental conditions, so change the nutrient supply and carbon?
Yeah.
And a bit more investigation of the mechanisms that might play a role.
Yeah, that one would be the next step. So we already did something regarding different nutrients conditions, but I cannot tell you much because I mean already at this stage the experiments were long and they take something like 6 years. But that one would be the next stage. So I can obviously do something on the modeling point of view, but we would like to have experiments to be sure. So I cannot tell you much on that, but that's will be the next case. At least changing nutrients. I don't know what's the plan for other kind of abiotic conditions, but for sure very interesting to be changed and see.
Let's thank the speaker again. All the speakers this session. So we will have a break now and we meet again 11 o'clock.